Today I'm going to talk to you guys about the eukaryotic cell. And right here I have a simple drawing that I did of a eukaryotic cell that displays some of the organelles that you might find in a typical eukaryotic cell, focusing on more of the organelles that you would find in a human. And I'm going to just name a couple of them and point to them on the diagram before we get into any detail about them. So right here you have the nucleus, and this dark region right here is called the nucleolus. Then you have ribosomes, which also you'll find ribosomes on the endoplasmic reticulum, but only on one type of endoplasmic reticulum, the rough endoplasmic reticulum. There's two types of endoplasmic reticulum, the rough and the smooth endoplasmic reticulum, and the only main difference is that the rough endoplasmic reticulum has ribosomes embedded in it, and the smooth does not. The next organelle I want to highlight is the Golgi apparatus, which looks a little bit like a stack of pancakes. And then you have the cell right here is pinching off to form a little pocket, and that it's going to make a little vesicle. And inside that vesicle, you're going to find digestive enzymes called lysosomes, which will break down the foreign material that the cell took in. Up here, you have centrioles, which are a type of microbody that help during mitosis or cell division. And then over here, you have mitochondria, which is the powerhouse of the cell and is the main organelle for metabolism in a cell. A couple organelles that I don't have pictured here are chloroplasts and centrioles, and those are mainly found in plant cells. Okay, the first thing I want to talk to you guys about is the cell membrane. And the cell membrane is called a phospholipid bilayer. And it's called a phospholipid bilayer because it has two layers. And it's arranged like this, where you have head with two tails and another head with two tails, and they arrange themselves in a circle like that. Now the head is polar or hydrophilic, which means that it has a charge. And the tails are nonpolar or hydrophobic, and they have no charge. Now, inside the membrane of a cell, there's going to be lots of proteins embedded into the membrane. And the proteins have many functions. Probably the top two functions would be cell identification and transport of materials across the cell membrane. Right here I'm drawing a little picture of a phospholipid bilayer cross-section. And you're going to have proteins that will traverse the cell membrane and make a channel for molecules to cross across. Nonpolar molecules will pass through the membrane very easily, whereas polar molecules will not. Okay, I want to now talk a little bit about the main organelles that you'll find in a eukaryotic cell. The first organelle is the nucleus. The nucleus is a double bilayer membrane. That means that it has two phospholipid bilayer membranes, or 4 layers. It will have pores in it. So if we have a little circle to represent the nucleus, you're going to find little pores studded all around the nucleus, and that will let RNA, or ribonucleic acid, come out. The nucleus contains DNA, which has the code for life and codes for different proteins in our body. And there's also a section in the nucleus which is dark staining called the nucleolus, and that is where ribosomal RNA is synthesized by DNA. Okay, the next organelle that I want to talk about is ribosomes. Ribosomes are synthesized in the nucleolus by ribosomal RNA, and they are used to make proteins. They're like the workbench upon which proteins are made. They have two subunits, a large subunit and a small subunit. And each subunit is composed of ribosomal RNA and proteins. And they're found in the cytoplasm of the cell. So if you have a cell here and the nucleus here, you're not going to find the ribosomes in here. You're going to instead find the ribosomes in the cytoplasm floating around making proteins. And another place where you'll find ribosomes is on the rough endoplasmic reticulum. The next organelle I want to talk about is the endoplasmic reticulum. And like I said earlier, there's two types of endoplasmic reticulum. There is the smooth and the rough endoplasmic reticulum. And the only difference is the rough is studded with ribosomes and the smooth is not. The endoplasmic reticulum, the smooth, is mainly involved in lipid synthesis and detoxification. And the rough endoplasmic reticulum is mainly concerned with protein synthesis. And that makes sense because it is studded with ribosomes, which we said earlier are used to make proteins. Another interesting organelle is the Golgi apparatus. And the Golgi apparatus is kind of like the packaging and processing plant of the cell. The Golgi apparatus is made up of membrane enclosed sacs, and it's going to receive vesicles, and it's going to modify them. The contents inside it will modify and repackage them, and then distribute the vesicle to the part of the cell that needs it. It will mainly release vesicles to the outside of the cell, and those vesicles will then be released via exocytosis. Now, a little bit about vesicles and vacuoles. Vacuoles are going to be mainly found in plants and sometimes in fungi, and vacuoles will contain and hold water, and that's what gives plants their turgidity. And vesicles and vacuoles are mainly for transporting material. Inside vesicles, sometimes we will find lysosomes. And a lysosome are digestive enzymes that will break down foreign material inside vesicles. Sometimes the cell will, in a way, commit suicide by releasing all of its lysosomes into the cytoplasm. And that is called autolysis. which just means the breaking open of lysosomes. And the only thing I want to say about microbodies is that they contain enzymes that are useful for metabolism. However, the main organelle of metabolism is the mitochondria. And the mitochondria is the site of aerobic metabolism, or aerobic respiration. And it's going to produce a lot of adenosine triphosphate, or ATP for short. And ATP is the main energy source for our bodies. Like the nucleus, the mitochondria has a double phospholipid bilayer. It's going to have the outer and the inner. The inner one's going to be really folded to give it increased surface area. And one interesting thing about the mitochondria is that they contain their own DNA separate from the nucleus, and they're able to replicate via binary fission. And the last thing I want to talk about are cell walls. And cell walls are similar to cell membranes, but they provide mainly protection from drying out. And so you're not going to find them in humans. You're going to find them instead in just plants, fungi, and protists. And that does it for the eukaryotic cell.